Anyway, welcome everyone. I am Adam Lawrence, president of the OA chapter. Thank you all for coming. Dr. Duncan, appreciate your coming tonight and presenting. A couple of things before we get started, and I'm going to hand it over to Sherry, our vice president. Again, if you'd mute your microphones, that way we can have Dr. Duncan give the presentation. There will be time after the presentation for questions and answers, so hold until after, please. There were some questions that were sent over when people registered. Those have been given to Dr. Duncan, so hopefully she can cover them. And if not, then maybe we can get them in the Q&A. Again, I think that's it. I'm going to turn it over to Sherry, and thank you all for joining.
Hey, everybody. Yeah, thank you, everybody. I think we have a really good, exciting speaker tonight, and I think we're going to all learn a lot. In the meantime, we've got some things coming up that if you either want to make a mental note or make sure you look for your e-blast in your emails, also social media. In July, we have a really exciting event. It's going to be our Vision Walk kickoff event. Not actually Vision Walk yet. Vision Walk isn't until September 13th. But this is to get everybody really excited about Vision Walk. It's going to be in person. And you're going to learn all about Vision Walk, teams, how to even create a team, the best way to fundraise, anything and everything you wanted to know about what's going on with Vision Walk. But the cherry on top of this meeting is we have a very special guest. Her name is Priscilla Gagne, and she is a 3-time Paralympian in judo. She has a silver medal, and she is visually impaired as well. And so she's going to give us a demonstration, judo demonstration. And then in addition to that, we are serving lunch. So be sure to watch for that email. It's going to be a really great event. It's taking place at Guide Dogs of America in Stillmar. In August, oh my gosh, another amazing event. I don't know if you've all heard about the Ray-Ban Meta smart glasses. Maybe friends have them or you've read about them. Well, we are doing a virtual event, so it'll be on Zoom. And we have a representative who's going to share with us everything we need to know about these glasses, how they could be helpful for both visually impaired, blind, and sighted. So it's for everybody. That is going to be August 20th. And then last but not least, just through September, because we have more in the works, is September 13th, lucky number 13, easy to remember, is going to be Vision Walk. And Vision Walk is at the same place it's been for the past few years, beautiful Santa Monica at Crescent Bay Park. And it's a really nice day, easy walking path. We even have shortcuts for those who don't want to do the walk. And there's going to be a vendor village where you could learn about all sorts of different products, some snacks, some breakfasty snacks. So it's going to be really great. And I hope you all can make it. So that's what we have lined up so far. And there's more coming. So let's get on to this event. That's why you're all here. And I'm going to turn it over to Monica, who could introduce who we're all going to listen to tonight.
Thank you, Sherry. Hi, I'm Monica. Thank you everyone for being here. I am extremely happy to have the opportunity to introduce Dr. Jacque Duncan. She's the chair and distinguished professor of ophthalmology at the University of California, San Francisco. She also serves as the chair of the Scientific Advisory Board for the Foundation Fighting Blindness, and also as the co-chair of the FFB Clinical Consortium Executive Committee. How does she have all the time? Dr. Duncan completed her medical training and residency at UCSF, and she went to University of Pennsylvania for her medical retina fellowship. Then she returned back to UCSF as a faculty in 2000, and she was appointed as the chair of the Department of Ophthalmology in 2022. She has expertise in the diagnosis and management of patients with retinal degenerations, including age-related macular degeneration, retinitis pigmentosa, cone rod dystrophy, and Stargardt disease. Her research focuses on identifying imaging technologies to better evaluate retinal changes in disease and in response to emerging therapies. On top of all her remarkable accomplishments, Dr. Duncan is also a truly wonderful person. She's kind, thoughtful, and deeply committed to the patients and the community she serves. I'm so pleased to have her join today. So please join me in welcoming Dr. Jacque Duncan for her talk.
Monica, thank you. What a kind and generous and very nice introduction. It's a real pleasure to be with you all virtually tonight. I'm coming to you from Northern California in my office at the University of California, San Francisco. I see patients every Tuesday. I see patients with inherited retinal degenerations and age-related macular degeneration and some other things too. But mostly my passion is to try and take care of patients with inherited retinal degenerations. And that's what I've spent most of my 25 years here on the faculty at UCSF working toward. So it's been really a wonderful opportunity to get to know a number of people who are affected with these conditions, family members of people who are affected with these conditions, and importantly, also brilliant scientists who are really passionate about understanding the problems and understanding them at a very basic level so they can think about ways that treatments might be designed that would help people retain their vision and maybe in the future sometime regain some sight. So I could talk about lots of things. I received your list of questions. I thought I would start with just a quick sort of summary of kind of where we are with clinical trials, which is a long ways from where we were when I started in this field 25 years ago. But there's a lot of clinical trials underway for patients with different forms of inherited retinal degenerations. And I'll give you kind of a brief overview today of some of the types of trials that are ongoing. And we've had some success, and then there's always lots of work to continue to do. So maybe today I'll talk a little bit about, I'm not going to share any slides because I realize not everybody can see the slides. So I'll just talk. And I'll try to talk slowly, and then I'll answer the questions that Monica sent to me in advance so I can try to address the questions people thought of in advance, and I'm happy to answer questions that come in to the chat as well that might be stimulated by anything I say today. So retinal degenerations are a very complicated, wide-ranging group of diseases. All of them share in common that they run in families and they're inherited. They're caused by changes in the DNA or in the genes that are present usually at birth. They don't always cause vision problems at birth, though. Over the course of time, they all share in common the progressive loss of the function and eventually the loss of the cells that help us see. And some of the retinal degenerations affect night vision first. Those are called rod or rod cone degenerations. Some of them affect central vision and daytime vision first, and those are called cone or cone rod degenerations, or sometimes macular degenerations. But all of them share in common two things. They are caused by genetic changes, and they cause progressive vision loss. The other thing that's kind of interesting is they also, similar genetic changes can cause vision loss in some animal models or in models that are like, we call them organoids, which are where you take blood or a skin cell from a person and turn it into like a little tiny eyeball. And that helps us study the disease and how the genetic change causes the vision cells not to work properly and not to survive. So the nice thing about these models is it helps us, one, understand the disease, and two, try out therapies that might be potentially helpful before we put them into people. So sometimes there are animals that are born with the same kind of genetic changes that people get that also cause the animals to lose their vision. And there's an example of a line of dogs that had a genetic change in a gene called RPE65. And that was a very uncommon cause of retinal degeneration in people as well. It caused early onset severe vision loss in little children or sometimes even babies, as well as in dogs. What was kind of interesting about that gene is that, as the name would imply, it was a gene that caused, a gene that was expressed in the cells under the retina called the retinal pigment epithelial cells, or the RPE cells. Those are not the light sensing cells. Those are kind of partner cells that keep the vision sensing cells alive and working well in partnership. So by identifying a form of retinal degeneration that doesn't affect the vision cells themselves, but instead affects their partners, that was a way to treat disease and cause vision actually to improve and to protect those vision cells and keep them alive longer. And there was a dog that happened to have the same kind of vision loss. So this was really somewhat fortunate in some ways, because it gave us an opportunity to understand the disease in a model and even try a treatment where we replace the gene, not me, but where scientists replace the gene into those cells. And they found that the dog saw a lot better. There was a very famous dog called Lancelot that actually went to Senate, the US Senate, and actually advocated for this treatment to be approved by the FDA because it helped the dog to see better when it was treated with replacement of this missing gene. So then fast forward a little bit, and they actually delivered the same kind of gene replacement to people with this kind of retinal disease, and found that the people also saw better. And this was super exciting. This was like the first time in all of medicine that it had been possible to actually replace a missing gene. Is anyone asking a question, or they just, if anyone has a question, I'm happy to address it, but I'll keep going if not yet. Okay, I'll keep going. So anyway, in people treated with this gene replacement, RPE65, they went from having very severe vision loss as little children to actually gaining back some vision. And the way they showed that this worked was by designing like an obstacle course called a mobility test, which they tested people's ability to walk around in different levels of light. And this was really exciting because they were able to show that the people that got this treatment were better able to walk through an obstacle course in dim light than the people that didn't get the treatment. And this is the first time they were able to use that test to show that people got better in response to replacement of a gene, and it led to approval, FDA approval of gene replacement. So this is the only real, in all of medicine, the only approved gene replacement therapy for any kind of inherited disease. And it turns out that it was successful in people with an inherited retinal degeneration caused by a gene called RPE65. So that was exciting. That led to a lot of companies to say, we want to develop treatments for our gene. We want to develop a similar kind of an approach for a different gene that we can replace if the gene's missing or it's not being expressed correctly. And so a lot of trials began and are still underway. And some of those trials have been shown to be safe. And some of them have shown some inflammation, but there have been obstacles kind of that have been identified and overcome as time went on. So there's a lot of gene therapy replacement kinds of trials underway. And as time goes on, sometimes there's genes that don't fit into the same kind of delivery vector. It's called a vector, but like a little delivery package that the RPE65 gene worked in. So there's other kinds of approaches where people try to skip over a mutation, and that's being tried for a very common cause of retinitis pigmentosa caused by a gene called Usher2A, as well as another kind of early onset retinal degeneration caused by a gene called CEP290. So they're trying to skip over a changed part of the gene using RNA, actually. Those trials are beginning to enroll patients right now and in the summer. So if you happen to know anybody with either Usher2A or CEP290, those are trials that are currently enrolling patients. A common cause of X-linked retinitis pigmentosa was treated with gene therapy and actually looked like a pretty promising result in a number of trials. And we're working with the investigators to try and convince them that this is important enough disease to try and keep developing that treatment in combination with a number of the scientists as well as Foundation Fighting Blindness to try and move that therapy forward through phase 3 trials and hopefully FDA approval. And then the last couple of things that sometimes people have tried to do for gene replacement include, you might have heard about CRISPR gene editing, where a protein goes and actually kind of identifies a genetic change and fixes the change and kind of cuts and pastes the changed spelling of the gene to make it spell correctly so that a normal protein is produced. So CRISPR was also used for the first time in people with CEP290 related mutations, and that gene approach was actually also very successful and resulted in a publication about a year ago. So that's another potential way of doing genetic gene therapy, gene replacement for people with inherited retinal degenerations. So those are a bunch of different approaches. For some people, the gene is too big, or the gene doesn't fit into the right kind of packaging. So we have to try other kinds of approaches. And then if you happen to have a dominant form of retinal degeneration, then there's other kinds of things that have to be studied, because you can't just replace the gene. So there's another trial underway for people who have a form of dominant retinitis pigmentosa caused by a gene called PRPF31. And this is an interesting one where the genetic change, the amount of vision loss is not caused by the changed gene, but actually by the expression of the healthy gene. So they're trying to deliver a different protein that will improve expression of the healthy gene to hopefully overcome the vision problems that ensue when people have PRPF31 related retinal degeneration. And that study is also getting ready to enroll patients. And finally, there's a couple of non-specific kind of approaches which aim to just keep cells alive longer. And a couple of those include a growth factor called rod-derived cone viability factor, which is being delivered using gene therapy to just try and keep cells alive longer. That's by a company called Sparing Vision. And then there's also ones that are just non-specific. They could maybe help everybody by reducing the oxidative stress in the eyes. And that's using a compound called N-acetylcysteine, or NAC. And this one is being delivered like an Alka-Seltzer tablet, which you dissolve in water, like a fizzy drink that you drink twice a day. And that's been being studied in the United States and other countries. They just completed enrollment of over 450 people with rod cone degeneration. Those patients will be studied for about 4 years. There's a related compound called N-acetylcysteine amide, or NACA, that's being studied in Australia in patients with Usher syndrome. And that should be releasing results pretty soon to see if that's going to move to the United States for additional therapy, additional clinical trials. And then finally, kind of the holy grail for treatment of people with retinal degenerations would not be simply to attack the disease early and slow down vision loss, but actually to bring some vision back. And some approaches that are being studied to try and do that include an approach called optogenetics, where you put a small molecule into the eye that responds to light. And it makes parts of the eye that aren't affected by the retinal degeneration become light sensitive, to try and replace the missing vision cells with new kinds of vision. So there's a number of companies that are trying to develop that type of an approach. One is called Nanoscope, one is called Ray Therapeutics. There's a number of different ones that are reporting that they're able to bring some vision back to people with a vision loss. Other things people have tried are prosthetic chips, little electrodes that deliver flash electrical impulses to the retina in response to light to restore some sight to people with vision loss. That was studied by a company called Second Sight for a number of years, and actually reached approval on a humanitarian device exemption back in about 2017. So there's some people who still have Argus 2 chips in their eye that flash lights in response to light from a camera that people wear in a pair of glasses. Then the last thing is 2 more things. Stem cells are being used a couple of different ways to try and release proteins that'll keep the vision cells alive longer. That's a company called J-Site is working on that. The dose, Laura Wild, the dosage of NAC is actually 1,800 milligrams twice a day. Thank you for asking that question. It's a little different than what you wrote there. So stem cells are being used to release proteins that are meant to keep the vision cells alive longer, and someday might be able to replace vision cells. So far, they have not been successful in kind of connecting to the remaining wiring that gets the information from the eye to the brain, but that is another potential way to restore some sight. And then lastly, there has been a big effort in the last year or two to try and reproduce some vision in people with vision loss from lots of different causes by transplanting the eye. So there's a lot of people who get a cornea transplant if their cornea gets scarred, or they maybe get a kidney transplant if their kidneys don't work. Well, a whole eye transplant has been really very challenging to think about, because the eye is kind of an extension of the brain. And so to replace it requires first that the eye not be rejected by the immune system, but more importantly, that the new eye connects up with the rest of the visual system so that it can send information that the patient can see and perceive and understand. So the United States government created a division called ARPA-H that has invested millions of dollars that were competitively awarded to 3 groups that are trying to develop whole eye transplant. And their goal is to transplant 6 eyes in the next 5 years at a number of different sites. So if that were successful, that might be another way to restore sight to people who have lost a lot of vision. So there's lots of different ways that people are working to try and treat these diseases. If you have RP65, right now we already have gene therapy available for patients with that disease. It's a very uncommon form though, but that success stimulated and inspired a number of other trials that are currently underway that will either slow down vision loss or may be able to restore some sight to people who have lost a lot of vision. I can stop now and answer some of the questions that you guys had submitted, or I can talk about other kinds of trials. There's other ones I didn't mention yet. There's lots going on in the field and it's a very exciting time.
No one has a question. Okay, go ahead.
Oh, no, I was just gonna say, Jeff, what do you think?
Oh, no, I was just gonna ask, great information, really appreciated. This has been very informative. Was just gonna ask, you know, some of these trials and different things that they're working on. Was just gonna ask if there's been any sort of side effects, or if there are any with these sort of things, and what they might be, to look out for.
I'm so glad you asked that question. And that's really, really the thing we always think about when we do design a clinical trial. The first step is always safety. The first step is like, this has never been in a person's eye before. So usually in a phase 1 study, patients who are candidates for those trials have very severe vision loss, and they receive a treatment to see if it's going to cause any harmful side effects. That's for safety. And then the phase 2 study allows them to increase the dosage and see what the largest dose is that won't cause a harmful side effect. And so some of the side effects that have been seen with gene replacement include inflammation. So, and that might be because if you put a gene into an eye that never expressed that gene before, that new gene product might be recognized as a foreign protein that could cause inflammation, or the packaging to deliver the gene might cause inflammation because they're usually viruses that have been sort of gutted of any of their viral material and replaced with a gene that you want to express. Or it could cause inflammation as a result of the surgery to deliver the treatment. So there's lots of side effects that can occur. Sometimes people have seen scarring under the retina. Sometimes they've seen inflammation inside the eye that can be treated with steroids. The other kinds of side effects that people sometimes experience have been, sometimes people who've had to have surgery can get a cataract or can get swelling in the eye. There's even a risk of retinal detachment. So there's lots of side effects that can occur. If we think about some of the pill ones, the ones that you take by mouth, like that NAC, that's been pretty well tolerated. It's actually FDA approved for other kinds of diseases, but sometimes that can cause stomach upset. It can sometimes cause people to not like taking the medication. They think the taste is bad. So there's lots of different kinds of side effects that can range from limited to the eye to more extensive throughout the body.
Oh, that's very helpful, and actually that's a good segue to one of the questions in the chat, which is it dosage for NAC still at 2,000 milligrams in those studies?
So the one that's being studied right now is 1,800 milligrams twice a day. So it's a little different than what the person who asked the question had thought.
Got it. Moving on, there's a few questions in here. I want to try and get to them all. Are there any trials going on for myopic degeneration?
There are. Myopia is a global epidemic. There's actually a huge amount of myopia affecting not only the United States, but globally. It's actually a really big problem in Asia. And so there's a lot of people trying to develop treatments for myopic degeneration. And there's a range of therapies being investigated that range from different kinds of light exposure to different kinds of contact lenses. If you're thinking about preventing myopia, little children are being given special glasses that have focus in the center, but not focus on the edge, being given drops of atropine to prevent them from focusing at near. And then someone actually asked in the next question about infrared treatment. Some people for myopia are being recommended to have low level red light. Other people for myopia are recommending blue light, thinking that like being outside in daylight conditions with short wavelength light might be helpful. Truly, I think there's a lot of controversy about what the right treatment for myopic degeneration would be. Other things that people have tried are like cross-linking to try and keep the eye from growing, different kinds of surgical treatments to try and keep the eye from, stretching as it grows. There's so much going on for myopic degeneration. To my knowledge, I don't know that any of it has been shown to be perfectly effective, but certainly it's an area of active research.
You kind of touched on the infrared treatments. Is there anything else you'd like to add to that?
I'm happy to talk a little more about this, but I don't know as much about it as maybe I should. But there is a treatment being investigated for age-related macular degeneration, which optometrists are actually able to give to people right now for a price that I don't think insurance will cover. That's been being used to try and reduce the amount of drusen or bumps in the retina, under the retina, for people with macular degeneration. So that is one new red light infrared kind of approach. I am not sure if it is or is not a good idea for people with retinal degenerations. I haven't seen any studies that suggest it's not harmful and might be helpful. The thought is that the infrared treatments cause some kind of low level damage or inflammation that incites a healing response that may be helpful for some kinds of retinal degeneration. I don't personally recommend it for my patients with retinal degenerations because I don't know enough about what impact it has on delicate photoreceptors that have genetic changes which cause them not to work properly or survive.
Great. So just a couple more here, and I think this is from the same question, or the same person, I should say. What to do when one's gene has not been determined yet? I think that's a great question, and and then the second part to that would be, is there any trials or treatments when it's been unidentified, which maybe is no, but if you could expand upon that a little.
Absolutely. Even in 2025, with really sophisticated genetic testing, when over 300 different genes have been associated with inherited retinal degenerations, even with that best testing that we can do now, about 30 to 40% of the time, if somebody with retinitis pigmentosa, who has nobody else in their family affected, has genetic testing, we are not able to figure out what gene is responsible for their vision loss. That's because there's probably a lot of genes we haven't discovered yet. Some people with that condition may have something different. They might not have an inherited disease at all. They might have some kind of inflammatory autoimmune form of retinal degeneration. But it's also true that there's a lot of people, even families with retinal degeneration, where we can't find a genetic cause as of 2025. So for those types of situations, non-specific protective kinds of approaches are being investigated. One example of that is the one I mentioned a little earlier, N-acetylcysteine, or NAC, is a non-specific antioxidant approach, which isn't going to affect the actual genetic mutation causing the vision problem. But the thought is that when there's a genetic problem, the vision cells undergo degeneration, and that causes extra oxygen to be around. More oxygen is being delivered to the eye than the vision cells are taking up. And that extra oxygen is interacting with light and causing reactive oxygen species. And that leads to oxidative damage, which is felt to accelerate the process of retinal degeneration. So the thought would be, if you could reduce the oxidative damage, one good way to do that is not by smoking. And another one is to reduce the amount of sun exposure by wearing sunglasses and a hat. But the thought would be, if you were to give a powerful antioxidant, in this case, N-acetylcysteine, you might be able to suck up some of those reactive oxygen species and prevent them from causing damage. And in animal models, that has been shown to be the case, which is why they conducted this large, multi-center, randomized clinical trial to see if N-acetylcysteine is also helpful in reducing oxidative damage and helping improve, preserve the vision cells in people with rod cone degenerations. So that's one of the things that are underway for people with unidentified genes. Other things that might be helpful include this one I talked about, rod-derived cone viability factor might be helpful for people with a wide range of mutations. Currently, that trial is trying to understand if it's helpful or not. So it's limiting the number of people that are enrolling to people with 3 specific genes, rhodopsin, CNG A3, and CNGB3, I think, CNG A1 and B1. We're not a part of that one, so I'm not sure, but there's 3 genes that are being enrolled in the Prodigy study, and that's moving forward into phase 3, I think. But it might be helpful for people with other kinds of degeneration as well. And then the other thing about people who don't have identified genes, any of the restorative therapies I mentioned could potentially be helpful. So things like prosthetic chips or optogenetics or stem cells like J-site, those don't care which kind of genetic form of retinal degeneration a patient has, and they might be helpful for any number of forms of retinal degeneration.
Hey, Jeff, may I ask a question?
Absolutely, Don, go for it.
So I was up at Stanford not long ago, and there's some people up there working on artificial retinas. Andrew Huberman interviewed one of them. I can't remember his name. Are you familiar with what's going on there, and do you have any hope for that?
Sure, yes, I am. The doctor who he probably interviewed is named Daniel Palanker. And he has invented a chip. It's one of these prosthetic chips I was just talking about that would sit underneath the retina and help deliver little electrical impulses to the eye in response to flashes of light. So they, I think, have been investigating patients with age-related macular degeneration and Stargardt disease as a first step. But it certainly may potentially have opportunities to improve vision in people with other forms of retinal degeneration, another approach that would not depend upon a particular genetic mutation being known.
Is Stanford really doing the best work, or are the other places doing the same thing, maybe other countries?
To my knowledge, the trial is not enrolling patients at Stanford. I think the investigators that developed the treatment are at Stanford, but I think the patients are being enrolled in Paris and Pittsburgh, if I'm not mistaken, for that trial.
Okay.
Any other questions? I have a few more in the chat, but I want to have some opportunity for anyone here who might want to actually ask the question. I'll leave a little bit of time here, and then I'll move on to some others in the chat.
Yeah, I'd like to ask a question. Can you hear me?
We can.
Yes.
Okay, this is Marcia Walensky. Hi, Dr. Duncan. My daughter, Jessie Walensky, actually saw you a couple of years ago when she moved to San Francisco. And through the testing, it's amazing, but you found her gene, the PRPF31.
That's one of the ones that we're just about to embark upon clinical trials for. So have
Right.
her
And I
come
was,
see me again.
yeah. And so I was talking to Ben Shaberman because I listened to his webinar a few weeks ago, and he told me about, well, he spoke about the trial in Oregon that they're doing for that trial, where they're doing injections into the vitreous.
Yeah.
Can you speak about that?
Yeah, thank you for bringing that up. That is one of the genes that causes autosomal dominant retinitis pigmentosa, and the degree to which the person's vision is affected is, in that case, determined by not the genetically changed copy of the PRPF31 gene, but the other copy. The amount that is expressed determines how severe the vision loss in patients with that form of inherited retinal degeneration. So this company called PYC Therapeutics is delivering a different gene called C03 to try and ramp up the expression of the healthy gene. So it's an interesting approach because it is gene therapy, but it's not trying to replace the gene that is affected in this condition. It's actually trying to improve the expression of the healthy copy of the PRPF31 gene. So it's a novel approach, and yes, we've been enrolling patients into the natural history study of that trial for several years now, and certainly they're beginning to treat patients with that C03 replacement.
So would Jessie at any point could be part of that trial?
Probably. I haven't seen her in a little bit, but certainly have her come see me and we'll have a conversation about it.
Terrific. Thank you, Jackie.
You're welcome. Nice to hear from you.
Yeah, thank you.
Thanks for tuning in tonight.
Any other questions before I move on to a couple more in the chat? And then obviously I want to give you a little bit more time in case you want to talk about anything else.
Sure. There certainly were there's a question in the chat about Alkeus. So I understand that Alkeus, which developed Gildeu retinol, Gildeuretinol, which is a form of deuterated vitamin A. It's a kind of vitamin A that doesn't incorporate into the photoreceptors the way regular vitamin A does, has been shown through phase 2 and now phase 3 studies to be protective against progressive vision loss in people with ABCA4 related Stargardt disease. They've reported that they've seen really promising results in phase 3 studies, and they are seeking to get FDA approval maybe in the coming year. So I have hope and optimism that it might be helpful within the coming year. There's a lot of other trials underway for Stargardt's too. There's one company called Belite Bio. These are different ways of taking medications by mouth to interfere with the way vitamin A is processed by the eyes. And they're all called visual cycle modulators. So certainly the Alkeus trial has made the most progress towards FDA approval, and I'm hopeful that they will be successful. That would be great. Someone asked about PROM1. I don't know of any clinical trials for PROM1. Pretty recently identified form of inherited retinal degeneration that I don't know if there's any specific trials for that one, but there may be non-specific trials that might be appropriate for some people who have forms of PROM1 related retinal degeneration. Someone asked if gene testing was a special test not covered by insurance. Yeah, great question. So fortunately for this community, Foundation Fighting Blindness has invested over $5 million in trying to allow people with inherited retinal degenerations to have really comprehensive genetic testing at no cost to them. This is through the My Retina Tracker registry. Because it was such a successful project, they recently reduced the number of genes that they're testing for, but it's still, if you've not had genetic testing and you would like to have genetic testing, please go see your inherited retinal degenerations expert and see if you can enroll in the My Retina Tracker registry and have genetic testing at no cost to you. Genetic testing is sometimes covered by insurance, sometimes it's not. It can be challenging to get insurance to cover it. I only end up ordering it through insurance if it's a gene that isn't covered by the My Retina Tracker panel. So for example, patients who have a different kind of disease like Stickler syndrome, or there's a couple of other, like albinism. There's a couple of things that aren't on the My Retina Tracker panel for when I go and order genetic testing, and it is hard to get sometimes insurance companies to cover it. But certainly important, very important part of standard of care that everybody with a retinal degeneration should have genetic testing. Even if they can't figure out what your gene is, you should at least have it, because if they do figure it out, you may be eligible for different kinds of treatment trials. There may be implications for other people in your family. It will give you information about kind of what form of degeneration you have and what to expect in the future, and whether there might be treatments on the horizon for you. And then lastly, someone has retinopathy of prematurity. So the Foundation Fighting Blindness has a mission that is really dedicated to studying inherited retinal degenerations, including age-related macular degeneration that cause progressive photoreceptor degeneration. Retinopathy of prematurity is a disease that causes retinal vision loss at very early stages, but it's more a vascular disease. In some ways, it's kind of similar to diabetic retinopathy or branch retinal vein occlusion, and it's a very early onset form of disease where, in the presence of too much oxygen, the retinal blood vessels don't develop properly, and it can cause vision loss if it's not treated as a baby. It really has to be treated when the people are newborn babies that are premature babies before they're even a couple months old. So depending on what the complication has been in your eyes, what's new now is we treat babies with different treatments that have been much more effective than they used to have in the past. One is by managing the amount of oxygen that they're exposed to. Another is by putting proteins in their eye that we use for people who have age-related macular degeneration and diabetic retinopathy to slow down the growth of those blood vessels and make them more normal so we can prevent vision loss. For people with really advanced vision loss who are adults already, the most promising kind of approach for that kind of disease might be this whole eye transplant that I was talking about a little earlier. It might be the kind of thing that a patient with advanced retinopathy of prematurity might benefit from.
Any other questions?
There were a couple that were sent to
Oh,
Monica. Oh, sorry, go ahead.
Oh, Millie looks like she's trying to unmute herself. Is someone able to unmute her?
Monica, did you have any questions yourself that were submitted or no?
Oh, no, so I submitted that to Dr. Duncan. Do you want to go through some of the questions, Dr. Duncan?
Sure, I'm happy to do that. So Monica sent me a list of questions that were submitted in advance. Someone asked if there was currently an effective treatment for retinitis pigmentosa. And the one that's been approved by the FDA is for people who have retinal degenerate early onset retinitis pigmentosa associated with changes in a gene called RPE65. That one has been approved. There are certainly clinical trials that suggest gene replacement for retinitis pigmentosa caused by changes in the gene called RPGR that causes X-linked retinitis pigmentosa look really promising, but have not been approved by the FDA yet. Retinitis pigmentosa associated with changes in USH2A are being studied in clinical trials with this exon skipping kind of oligonucleotide approach, antisense oligonucleotide approach through the LUNA study. And there are the sparing vision studies where they're using raw derived cone viability factor might show to be helpful for people with retinitis pigmentosa. This approach for PRPF31 with C0.3 might be helpful for that form of retinitis pigmentosa. And then non-specifically, things like NAC might be helpful for people, like antioxidant therapies might be helpful for people with retinitis pigmentosa. So a lot of work underway. Nothing has been proven effective yet, except for RPE65 gene therapy. And then is there any research on infrared therapies? We talked about that. Has the retina successfully been transplanted yet? Not yet, but the hope is that this whole eye transplant will be an effective research experiment that leads to treatments in the next 5 years. Stay tuned. And when would it be available to people in the general public? Well, first it has to be shown to be safe and helpful. And so I would expect, if it is successful, it will be available shortly after, maybe within a year of being shown to be effective. Treatment for Stargardt disease involves right now avoiding extra vitamin A and sunlight exposure. But as someone asked about, the Alkeus trial might result in an FDA-approved drug later this year. Someone has their hand up. I can stop talking if you want me to. And there's more in the chat.
Greg, do you want to go ahead? You have your hand up, and then we have a couple more in the chat.
Thank you. What are some of the timelines for these treatments that you're talking about that are in trials?
Well, that's a great question. It depends a lot on the duration of the trial and the stage at which the compound has reached, right? So the Alkeus study has honestly been underway for probably 10 years. They've been studying it in different phases for many, many years. They finally completed the phase 3 study, and they're pretty close, they say, to receiving FDA approval. That one's probably within the next year. Other ones that are just getting underway, like the NAC attack study I mentioned, just completed enrolling over 450 patients, and that is a 45-month study. So we have to wait at least 45 months plus some time to analyze the data before we find out what the results of that study will be. Most trials are usually about 2 years when they're looking, and many are not yet in phase 3. Many are in phase 2 where they're trying to decide what the right dose will be. So if you have to be successful in phase 2 to pick the right dose to go forward with phase 3. Usually to determine if something's slowing down vision loss, the trial has to be a couple years to show a difference between the treatment arm and the not treatment arm. So probably 5 years or so for the things that haven't yet reached phase 3 trials, but that's sort of it depends. If it works really well and it shows a big difference, and something like RP65 shows visual improvement, then you can show a difference in a very short period of time. So it depends on lots of different things. There's a couple more in the chat. What currently looks most promising for AI PL1 cone rod dystrophy? That one was studied. That's a gene that fits into the vector that RP65 was delivered in at Moorfields Eye Institute in London, Eye Hospital in London. That was shown, like, had a lot of potential early promise. The challenge with that one is it tends to be very early onset. And so you have to intervene at a very young age to avoid to allow the brain to develop the ability to see. And so I don't know the status of that, but I know that that was investigated at least in very early phase studies at Moorfields. So stay tuned. Hopefully there will be treatments for AIPL1 at some point in the future. And the next one was about the recording. Would the recording be available? A couple more questions that were sent to Monica include about cone rod dystrophy. So cone rod dystrophy is a little bit less common than rod cone degenerations. There are certainly the most common cause of cone rod dystrophy are probably ABCA4 and RPGR, probably the two most common genes associated with cone rod dystrophy. Both of those are pretty common causes of degeneration, and treatment trials are underway for both of those if that happens to be the genetic cause for the patients that are asking about it. If not, someone's asked about PROM1, so that's a little less common, and so may require a little bit more time to develop treatments for those kinds of approaches. There's a company called that the Foundation Fighting Blindness founded called Opus Genetics, which is dedicated to trying to develop treatments, genetic replacement treatments for people with rare forms of retinal degeneration. So that might be the kind of thing that they would be able to investigate if there were strong animal data or preclinical data that suggested a treatment might be helpful. And then somebody asked about a specialist in Phoenix. The person I know in Phoenix probably is that I would recommend.
The charger for your phone.
Guy named Ben Bakal, Bakall, is in Phoenix, and he trained at Iowa and is a great expert in inherited retinal degenerations. Hopefully that's helpful. And lastly, the best place to find information about clinical trials, there's two places. One place is the Foundation Fighting Blindness website. So if you go to fightingblindness.org and type in trials, there's a wealth of information. Much of it has been curated for you by a guy named Ben Shaberman, who's incredibly talented at like distilling really complicated information into bite-sized, manageable, understandable formats. That's one good place to start. And then if you don't see what you're looking for there, you can always go to clinicaltrials.gov. That's where I go all the time to see what the latest status of clinical trials are. You type in the gene of interest or the condition that you want to learn more about, and it will list a number of trials underway. Many of those are not treatment trials. Many of them are natural history studies or just trying to identify genetic causes of disease. You should also not, a lot of things are listed on clinicaltrials.gov. That doesn't necessarily mean that they are supported by strong clinical evidence or scientific evidence. It's just telling you the trial's underway. So it gives you information about studies that are happening, but they haven't really been vetted for any kind of like whether or not it's something that would be recommended by the scientific community. So that's why I'd like to refer patients to the Foundation Fighting Blindness website first. And if there isn't any information there, then the clinicaltrials.gov is another resource that is more comprehensive, but not very well curated or vetted. Everything is up there that you have to kind of sort through by yourself. Other questions? I think we got through most of it.
There's a few. I mean, we never had so many questions. So I mean, this is great. But
I'm excited that people are interested.
Yeah, Monica posted a few more in the chat.
These questions came in after I sent you the list, Dr. Duncan.
Oh, okay. Okay. If the findings of a clinical study do not show the anticipated substantial advantage in treating a disease for which there is no known cure, what would be the requirement for altering the trial's endpoint? That's a really interesting question because, and that has been the downfall of far too many clinical trials, I'll be honest with you. When a study is designed, the primary outcome measure has to be picked by the people that are designing the study before anybody's enrolled. You have to design the study and decide what you're going to monitor, and you have to say what you think's going to be the thing you're going to follow. And you can have secondary endpoints too that you're also going to look at, but the FDA only cares about the primary endpoint. So it's very, very important to pick that primary endpoint based on natural history studies of people with the disease. You have to understand what the disease does to the vision and over what period of time it changes to know what the right endpoint that you expect to change over the course of your trial will be. And I will tell you, not infrequently, maybe always, people pick one that isn't necessarily the one that is going to change over the course of time and doesn't show. This is why trials sometimes look promising, look safe, but fail to meet their primary endpoint because the trial was designed in a way that picked an endpoint that maybe is one the FDA likes or one that the people paying for the study think is one like visual acuity or something that they know is something the FDA is happy to look at, but isn't necessarily one that's likely to change in the disease they're studying during the time period they're investing in, a couple of years. So more often than not, I think that I can't emphasize enough how important that question is. The Foundation Fighting Blindness Consortium spends a lot of time conducting natural history studies so that we really understand what the right outcome measures to measure and monitor and pick for the treatment trials are going to be. So then when somebody comes along with a really promising treatment, we can say, listen, if you look at this, I predict the people, if you enroll this kind of person, they will change by this amount of this amount over the kind of time that you're looking for. And either you need to design a trial that is really long to show change, or you pick a measure that is likely to change in the amount of time you're willing to study, and then you randomize your participants to either get the treatment or not. And hopefully the people that you pick are going to behave like the natural history studies, and people you enroll are going to be like the ones that have been studied before, show the same amount of change in the same amount of time, and you can see whether the treatment made a difference in that or not. So that's a super important point, and I'm glad you brought it up. I think we talked a lot about Stargardt disease, and then somebody was diagnosed with RP, but the appearance is choroideremia. The gene has not been identified with current testing. Yeah, so choroideremia and some forms of RP so RP is such an interesting disease because it can affect the cells that live under the retina, the RPE cells. It can affect the blood vessels that deliver blood to the retina, like the choroid, which is where choroideremia probably looks like, at least clinically, although it probably affects all the cells. It can affect the vision cells themselves and not look like much of anything on exam. And so it's because different kinds of genetic changes affect different cells to different extents and cause different appearances in the retina. And for hundreds of years, people called all of these diseases retinitis pigmentosa. Like they lumped them all together and gave them a name because the retina looked like there was inflammation and there was pigment. And then little by little, beginning as recently as 1990, genes were identified that were found to cause this kind of vision problem. And since then, 300 different genes have been identified, and there's probably 30% of people for whom we still don't know what the cause is. So it's a really complicated condition caused by hundreds of different types of genetic mutations. This is one of the reasons why it's been so hard to develop treatments, because it's actually not one disease. It's actually a whole bunch of diseases that look kind of similar when the eye doctor looks in there. And so we very simplistically lump them all together and tell people that we think they have this disease, and we might not know what the cause is at all. And so it makes it really hard to develop a specific treatment when we're not sure exactly what the cause is. So I guess the type of advancements are just maybe have genetic testing again in a few years, because new genes are discovered all the time. And maybe you might, depending on what is going on in your eye, be a candidate for a non-specific treatment like NAC attack or NACA if it comes to the US in the future, or just stay tuned, and there may be treatments available that are non-specific to restore some sight. Maybe that could be optogenetics, or maybe that could be prosthetics, or it could be stem cells. So lots of different avenues depending on what stage of vision loss you currently have experienced. And then someone said, how can someone contact, you talked about JSite, you going to clinicaltrials.gov is probably the best way to do that, and type in JSite under the sponsor, and you can learn whether there's, at what stage of patients they're enrolling right now, or what the progress of that research has been. And then how can someone contact Opus for treatments? Yeah, so Opus, I think it's called Opus Genetics, is a company that was created by the Foundation Fighting Blindness, and they're currently investigating a small number of genes that have been mostly like in partnership with people that want to support development of that gene, or in combination with preclinical data that suggests there might be a good treatment for that gene, are raising funds to develop treatments that are specific to particular genes. I believe you can learn more about it by going to the Foundation Fighting Blindness website and typing in Opus Genetics. It will probably take you right there, or there may be a website directly for Opus Genetics.
Can we go back to Stargardts for a minute?
Sure.
So my brother and I both have Stargardts, and I know many Stargardtians. And I heard in the Stargardt Summit that the ABCA4 gene is a large gene.
Correct.
And how are variants identified? I had late onset, fortunately, but are they identified with numbers and letters? How does that
Yeah,
work?
that's an interesting question. You're right about that. Everything you just said is completely true. The ABCA4 gene is very, very large. It's really, really big. And the way we talk about genetic changes is by so if you think about a gene like a word, so a word is comprised of a number of letters. And so the ABCA4 word is hundreds of letters long, whereas the RPE65 gene is like a very short word. And so the change in the word or the gene is defined by a codon. And so it is defined by the letters that are misspelled, if you think about it that way. So in ABCA4, there's a whole bunch of different letters that could be misspelled. There's a lot of locations in the big long word where you could accidentally have a typo. And it's even more complicated in ABCA4 because there's parts of the gene that don't even turn into protein that are called introns that can sometimes have changes in them that is not a usual way of causing vision problems, but in ABCA4 that certainly happens. So it's been challenging to identify all the genetic changes that could exist in ABCA4. It's also challenging to treat that big gene because it doesn't fit into the packages we usually use to deliver the gene. So there's lots of companies that are trying to find creative ways around that problem by there's a company called Ascidian that's trying to develop a treatment for the people who have changes in the first part of the gene, exons 1 through 22, where they're going to deliver a way to get sort of fix that part of the gene, or they're going to deliver a little bit of the gene and hope that that's going to overcome the part that's missing. There's another one called Splice Bio that's doing a similar kind of an approach. And there's people that are trying to do dual vectors where they put like half of the gene in one package and the other half in another and hope they both get into the same cell. The other problem with ABCA4 is there's a whole bunch of different changes. It's not like there's a couple that are common, but there's a bunch that are very uncommon. So unlike USH2A, where there's one area, exon 13 is pretty common. You can design a treatment for just that one particular form of genetic change, and it will be relevant for a lot of people. ABCA4 is less that way. There's changes all over the gene. And so it becomes a lot more challenging to develop treatments for that one because it's a long gene and it's too big to deliver the whole thing. And so people are trying to be very creative to outsmart that and just replace part of the gene or replace two halves of the gene and hope they both get into the same cell or skip over the part that's changed.
So what are the genes associated with age related? I've never heard anyone talk about that.
Yeah, there's over 20 different ones. The most common ones are complement factor H and HETRA1. And those are two. But the thing with the age-related macular degeneration, unlike inherited retinal degenerations like we've been talking about all night, it's not, those are called monogenic changes, where like monogenic diseases where if you have a change in one gene, you're gonna have a vision problem, almost certainly. ABCA4, lots of people have changes in lots of genes that put you at risk for macular degeneration, but they never develop a problem. Other people have one or two changes that cause very severe disease, and other family members with the same genetic changes don't have any problems at all. So it's called a polygenic or a multifactorial disease. It's not as it's much more complicated, less straightforward than people with inherited retinal degenerations caused by changes in a single gene.
My brain is starting to hurt.
Yeah, me too. It's 8. Shall we wrap it up?
I just have a really quick question for you, doctor. Kind of a different topic, sort of. You've got a patient who comes to you, they're either newly diagnosed or maybe they're coming to ask for a second opinion, or maybe even looking for a different answer other than I'm visually impaired and I might lose my vision. Do you have like I don't know if speech or routine is the right word, but something to kind of give them the understanding that, yeah, there is hope. There are a lot of different clinical trials, et cetera, but there are some things you might want to think about to prepare for the possibility that down the road you may not have sight. Something to maybe get them to maybe kind of understand.
Yeah, that's a really interesting question. And I'll tell you, the first time I meet somebody, if it's a brand new patient to me, that is not where I start, because I'll tell you, a lot of people, I don't go there first ever, because most people who come to me have been told they have a vision problem that's going to cause them to go blind. There's nothing you can do about it. Like that's 99.99999% of people that come to my office. So they've heard that message. And what I try to help them understand is that that is kind of the way people think about these diseases. But at the same time, compared to ever in the past, all of history, we have a lot greater understanding of the diseases now, and there's a lot more research being done, and there's been success stories. There's been a treatment that was FDA approved for people with one form of inherited retinal degeneration, which led to a lot of other trials, and there's a lot of progress being made. And I leave them with, there's so much we can do right now to help you. First of all, the other thing people get really worried about is that they're going to lose all their vision tomorrow. And I tell people that it's a very, very slow change, and it is fun. It is literally in your DNA. This is something you were born with. This is a part of who you are. You've had this your whole life. It's now becoming more of a problem such that it's affecting your vision, but you're not different today than you were yesterday, and you won't be very different tomorrow. So it's a part of who you are, and it's a disease that today we understand a lot better than we used to. And there's lots of hope that in the future there will be treatments that will slow down the rate of vision loss and maybe even restore some sight. So I tell them that first, and I say we want to do the best we can for you by learning what you have. And so we try to get genetic testing to try and understand what they have to the best of our ability. We do as much testing as we can to understand where their visual limitations are, because sometimes they don't know. They are not aware of where they can and cannot see things. And then I hook them up with low vision, even at very early stages. I introduce them to people who can help them with adaptive technology, which, like, you started off this meeting tonight saying that there's these new glasses, there's like new developments being made all the time with technology and different kinds of things that are really, really helpful to people. And so I think that that's the message I try to start with. I appreciate that it's important not to sugarcoat it and make it sound like everything's going to be totally fine. And I have, I'm going to wave a magic wand and you're going to be perfectly okay. But I am going to let people know that they are going to be okay, right? They've had this their whole lives. They've found ways to compensate so far, and they will continue to do so. And in the meantime, the research field is making great progress, and hopefully there will be different treatment options available in the future than there are right now.
Okay, thank you.
That is such a great approach. I just want to comment on that, because in all the appointments, certainly I've been on with Adam, no one's ever said, you've been this way your whole life. Like, this is who you are. And these are just things that you're going through. And that was like, I'm pretty impressed by that. Thank you.
Thank you of course, and I think sometimes people feel like, did I do something wrong to deserve this? Or like, should I have done something differently? No, this is who you are. The other thing to know is everybody, I want to tell you that everybody has something, right? This is your thing. Everybody has something, and they don't always show it and you don't always know it, but like everybody struggles with something or another. This is the thing you have, and you've had it all the time, and we're going to be there to help you get through it as best we can.
All right,
Wow,
well,
with
great.
that, that's a great ending.
Yeah,
I
that's
think,
terrific.
right there, right,
Thank you.
Jeff?
Yeah, I think that's the spot. Again, thank you, Dr. Duncan. Really appreciate your coming on and talking to us tonight.
It was my pleasure. I wish you guys all the best. I hope that we run, cross paths again. I see Monica all the time, it seems like. So if you ever want to share a message, get a message to me, you can tell her and she'll find me.
Thank you
you
so
so
much,
much,
Dr.
All
Dr.
right, it was a
Duncan.
pleasure. Take care, everyone.
Take
Bye.
care.
Bye-bye.
Thank you so much.
Okay, well, that was so great. Thank you again, Monica, for arranging that, and want to thank everyone for all those really thoughtful questions. I certainly learned a lot from that. So, sort of the last half of this meeting and kind of what we wanted to talk about is sort of a section that we call open forum. And this is just a time for us to kind of share our experiences, share our thoughts, share any tips and tricks that we may have. And so, just think of it as a way that maybe someone who's going through something similar to what you're going through, maybe didn't know this, or maybe we'll just find this out for the first time now. And your experiences, do matter, and they matter to a lot of us. So, with that in mind, It's kind of a sort of a starting opening question that we have and that I'd like to ask. And hopefully, if you feel comfortable in doing so, would love for you to share your thoughts and your experiences. And so the question I'd like to pose to this particular group is, what are the two most challenging issues you have faced as a visually impaired person, and how did you deal with them? So that's the question that I have. And, I'll be happy to go first myself and sort of answer that to kick things off. And then hopefully we'll get some other people that would want to chime in and sort of share their experiences. We would love to hear from you. So I'll start it off and kick things off. And for me, I would say that the two most challenging things for me were really mobility, and that went to, many different factors in that. And the main one really was driving at night. That was a big change for me, not to drive at night. And it was something that I felt like I was losing my freedom. I couldn't go to different places that I wanted to at night, different events, sort of had to before Uber and all those sorts of things, sort of always had to arrange things and figure out a way to do that. And for me, the biggest way now, it's a lot easier with Uber and that sort of thing. And I rely upon that a lot, but also just, with my family, with my friends, with my partner. That's really how I've tried to overcome that the best, is just sort of relying on people a little bit, who are willing to help. And there's always people out there that are close to you, that love you, that are willing to help and understand, sort of what you're going through. And so, that was a big challenge for me, and also just mentally, it's just sort of coming to terms. I think Adam was sort of talking a little bit about this, but sort of coming to terms with the fact that I just can't drive at night, and that's just no longer really something that's safe and an option for me. And once there was a little bit of acceptance on that front, it made sort of dealing with that challenge, a lot easier for me. And I would say that the second thing was really, me being here. I think one of the biggest challenges I faced was sort of feeling like there was a stigmatism about it, and sort of what the last thing that we talked about with Dr. Duncan, that just accepting that this is just me, and that there's nothing wrong with it, and that many people are going through it. So one thing I often had to early on in my life was sort of hide from it and not let people know that I even had anything, because oftentimes during the day or in certain situations you wouldn't know that I really had any sort of condition like that. So really the way that I overcame that was in large part, thanks to my family, my partner specifically. She really helped me out with this, who's on this call, in fact, and so is some of my other family members. But really was this foundation, actually coming to some of these meetings, and really seeing people that were going through the same things, sharing their experiences, actually made me feel comfortable talking about it. And I never was comfortable talking about anything like that before, was never really open about that before. And so it made it so that I am more comfortable now talking about it, and more, able to share my experiences, and just that this is a reality that I'm going through. So for me, in large part, the foundation really helped me, and these sort of community connection events that we're having, that was really one of the starts to sort of having me be able to open up and share, sort of that this is who I am, and this is my experience. And so those are my two biggest challenges really were from a mobility perspective, really, and just from coming to terms and being able to talk about this as a real thing, and share that. So anyways, with that said, hopefully we'll get somebody else that might want to share their experience if they're comfortable doing so. And we'd love to hear from you.
I could share some. My name is Bhushan. Can you guys hear me okay?
Yeah, we can hear you.
Yeah. No, definitely the mobility part, you hit it right. I'm actually legally blind, so I don't drive at all. And it's been pretty rapid degeneration over the past 5 years. But what I would like to suggest, actually, I mean, yeah, we all deal with that, and we'll come to grips with it. Like you said, Uber, Lyft, they're pretty good options these days. There was also, I don't know if I'm sure many people would know, there's also the county services, right, through the ADA Act access. which is not the best way to travel, but at least it's a way to travel, where the county services come pick you up and take you in a shared ride kind of environment. But the challenge I faced there was we just moved to a new location, and that service is tied to how the public transportation bus route works. And they truncated the bus route. So suddenly we had that service and we moved to this new place, and within a month, the service stopped, right? So a suggestion would be through this organization, if we can approach the legislators to see if they can help kind of solve the problem for not just the people who are blind, but a lot of people who kind of tend to depend on these services. And maybe they need to evolve that program and partner with Uber and Lyft, not just through the county transportation services, is one thought. The other, definitely, if we can put in our 2 cents of driverless taxis, that would be a big help for people like us.
Great suggestion. Thank you, Bhushan. I really appreciate that. Very good thoughts and tricks. And absolutely right. Sometimes it's really about making our voice heard. They always talk about, legislation and different things, cutting different funding for different things. And I think sometimes, we just have to make our voices heard. And so great thought there. Anybody else feel comfortable sharing any challenges that they have faced and how they've overcome them? Would love to hear from you.
I think we have Dawn.
Yeah, hi. I want to say that, I have macular degeneration, and as you guys know, some of you know anyway, I'm really into technology, and I have the Meta Ray-Bans on right now, and these glasses have been the best thing that I've found in the last 5 years in terms of helping me, read signs at a distance. I would have to go up to a sign and be 2 feet away from it. Now I can be, 25 feet away and read it through the glasses. So I'm looking forward to your talk on August 20th. That the glasses are now enabled with live AI, so I can have a conversation with my glasses. I can use it to translate from Spanish to English real time. They're fantastic devices. Google is coming out with glasses,
Yeah, I just
smart glasses soon, that are going to have a heads-up display in them, which doesn't help someone like me, but it may help someone with RP, for example, who still can see things up close. And then it's rumored that Apple's going to come out with a similar product next year. I really think that these smart glasses are the future, not just for people like us, but for everybody out there. It gives you added advantages and also gives you eye protection. So that's my 2 cents.
You, Don, always good to see you. Really appreciate you. So great tricks and good segue to a thing that we have coming up. So thank you for that plug there. Anybody else? Anybody else have any thoughts, any comments, anything you'd like to share?
There are also rumors that the Apple Vision Pro will act as a telescope that's being worked on. So that could be really cool for, watching TV or whatever, for those of us that have some retina left.
Any other thoughts? Don't be shy. I don't want to have to call on people directly. So,
I want to, am I unmuted?
You're good,
Yes.
Vicky. We can hear you.
Okay, this is Vicky. This was a great session. Thanks. I love the guest speaker. I was in line for J-Site for my doctor's been talking to me about it for 2 years, and it finally came to be. They called me. I went through the informed consent. This is kind of a crybaby talk. And then all of a sudden, 2 days before my appointment, they called me. They realized that I'm over 60. And they said, "Oh, oops, sorry." So it's been a real struggle, it was like so hard to hear that. And I wanted to ask the doctor, but I didn't get the chance. how do they determine the top age? You know, I still have quite a bit of vision, even though I'm over 60, over 65. But you know, I was like, it's a random number. And then my other question for you, Don, is do you think an iPhone would be better than an Android for
Yes.
this?
Sorry.
Okay. Okay. But anyway,
I
so
jumped the
that
gun, Don. I apologize.
that's okay. That was just my sad tale of woe that I mean, it's been 2 years coming and I had the appointment, and then they anyway, it was so sad.
Oh, we're
Well,
sorry to hear.
to answer your question, Vicky, I could talk about Apple products for the next hour.
You know what? I'm going
Um,
to email you, Don. I have your email and then we'll talk on the phone. How's that?
That's good.
Okay.
Vicky,
Well, we may line
it's
you
a
up
great
again, Don.
fun.
We had you
Sorry.
once before, so we may line you up again for another presentation. So
And
that would be.
I'd be happy to do that. I would
Yeah.
spend time talking about these glasses, but maybe I'll just fit right in on August 20th.
Yeah, Vicky, it's a great product, Apple iPhones. I mean, I've been using them for years and I don't know what I would do without them. And, you know, not to knock Android, it's, you know, it's an okay product. It's just, in my opinion, the Apple is got a better voiceover and, you know, interface. It's just, it's
Mhm.
easier, it's better. It is a
I'm
learning curve, no doubt about it. But
yeah.
once you get through the learning curve, I mean, it's just,
Well, you know, I use an iPad, so I'm familiar with iOS. I just have always had a Samsung phone, but I'm just thinking as my vision is slowly declining that maybe I want to swap over to iPhone.
Actually, I
I
think
think you should. I think you should.
I do too.
if,
Apple's come out with a cheaper phone. I mean, you can get a
Yeah, they
16E, it's
yes.
called, yeah, which is
Yeah.
entry
Yeah.
level, you
Yeah.
know, for $600. That's pretty good.
Right.
All the things they can do. I mean, you know, it's still got a few things they're working out with the AI and stuff, but I gotta tell you, I don't know what I would do without it. And just, I don't know, I think it's a great system compared to,
Yeah.
but
Yeah.
that's just my,
Plus,
if
everybody in my family has one. If I want to message them, you know, well, I can message them, but you know, the iMessage is so different. But anyway, yeah. So that's my sharing tonight.
Well, thank you
sorry to
for
hear about
sharing.
J-Site. You know, I don't know what to tell you. I think Dr. Duncan kind of alluded to that question and was asking about, you know, how do you, don't remember what it was. It was a long question, but I think she was saying something to the effect of that, you know, they have to have a certain amount of time to monitor the patients and stuff. So maybe the age is a factor in how long it takes to
But
monitor.
my argument for that is I'm a healthy 66-year-old. There
Well,
could be
absolutely.
a 30-year-old who's big and fat and not healthy
Right.
and who
Yeah.
qualifies. You know, that's
No, understand. I'm not defending it. I
yeah.
totally agree with you.
Yeah.
I don't understand why they're cut off either, but you know, I guess that's what there is.
Well, you know, Monica, before Bhushan has his hand up, and I'll get to him in a second. But Monica, if you could maybe relay that question to Dr. Duncan and just see, you know, if there's any sort of why there is an age and, you know, why it sort of cuts off there. And then we could sort of relay that to Vicky. We see Vicky a lot, and we can, you know, sort of relay that to her. So,
I can definitely do that.
Thank you.
Bhushan, go ahead. I see you have your hand up.
Sorry, I just had a quick comment. You guys are talking about this next session, and I heard the two people are big iPhone and iOS fans. I was wondering if, and maybe you guys have already planned this, so just confirming then, if you guys can just walk us through what is it you guys do on the iOS device that makes life easier for being blind, you know. I have not used iOS. I am an Android guy, and
Woohoo!
now I switched to the Google Pixel 9, and that has some very cool accessibility features, but I don't think I'm maximizing what it is offering. So it would be good to know what you guys are doing on the iOS, and maybe that's the plan for this next session you guys are talking about. But if not, then maybe we can have a sort of a tutorial of, you know, hands-on users in this group.
Do you want to take that one? Because I don't have an iPhone, so I'm kind of I can't really comment on that. But I would say that kind of answered one of my questions, before we were going to run out of time, which was we'd love to hear what presentations that you would like to hear, and sort of what you would maybe want to hear in the future. I know we have a little bit of our calendar planned out, but we would always love to hear from the community, especially this LA chapter, about what they're interested in hearing. And I heard from you that it would be something about technology, something about sort of walking through different iOSs and different systems and accessibility features. And I think that would be an excellent presentation. So I'll let Dawn maybe take that one, and we're running up on time here. So maybe have another 5 minutes for maybe sharing a little bit, and then we'll kind of do our closing comments and let you guys have back the rest of your night.
Well, iOS, I mean, if you can do something with an Android, iOS can do it, just as well. So where iOS is good is that if you get tied into the Apple ecosystem, and I spend most of my time on a Mac, and the phone and the iPad just tie in so beautifully with the Mac. And they also have a great 800 number for people who have vision or other accessibility issues I mean, I know a lot about this stuff, but I end up calling them, probably every 4 or 5 months with a question. Turns out to be a pretty stupid question, but I couldn't figure it out. So I just call them, and it's so easy to dial that number and get someone that really can help you with whatever is going on. I always tell people that if they're in a work environment, or if they have family or friends that are in Android or Apple, they probably should stick in that area, because having your daughter or your son or your brother-in-law, whatever, help you out, that really is worth a lot, whether it be an Android or Apple. So, but I happen to know a lot about Apple and follow them really closely. And so with having said that, I mean, that's about all I can say. But Vicky, you only look 45 to me.
Go, Todd.
That
Well,
was a joke.
anyways, it was really great having everybody here. Really great questions. Great to see everybody, and never hesitate, to drop us an email if you didn't want to share right now any, sort of presentation you'd like to see. But you can email us at ffblosangeles@gmail.com, and we'd love to hear any feedback you have for any future presentations you would like to see. With that said, I'm going to turn it over to Adam. For sort of closing comments, and we'll take it from there.
All right, thanks, Jeff. Great job. Thanks to everybody for joining tonight. We didn't announce at the beginning, but this was recorded tonight, and eventually will be on the FFB YouTube channel, and we'll get that information out through an e-blast if you want to listen to it again, if you missed something or you want to go through something again. Also, again, as a reminder, we do have our upcoming events. The Vision Walk kickoff with the judo presentation, July 26th. That is an in-person event. It'll be in Sylmar. Hopefully a lot of you will want to come and join and participate. We also have the smart glasses virtual presentation in August. Again, something that we hope everybody will be interested in, and of course, Vision Walk in September. I do want to thank all of you for joining tonight. We appreciate your supporting us by showing up, participating, attending these events. We try to put them out for you guys to cover different subjects that we think you'll be interested in, but Jeff's correct. We love to hear if you have something in mind that you'd like us to try and put together. We're always happy to try and figure it out and make it happen. And with that, I thank you again, and have a great evening.
Thank you, everyone.
Thanks, Adam.
Take care.
Thank you, everybody.
Thanks, Adam.
Good night, everyone.
Bye.
Bye.